Good morning, students. Today we will see the important topic membrane potential. Each and every cell of the body is having a membrane potential. So, what is the membrane potential and what are the factors affecting the membrane potential? All those concepts we will see. So, the base book and the reference book for this topic which I have chosen are Boron and Ballberg, Best and Taylor, Guyton and Hall, and Byrne and Levy textbooks. So first we will see the contents under which I will be covering this topic. Just a brief introduction and the basic physics of membrane potential. Membrane potential caused by ionic concentration and the Nernst equation, Goldman equation, resting membrane potential of a neuron, and active transport of sodium potassium ions through the membrane, and what is the origin of normal resting membrane potential, and finally how to measure the membrane potential. So at the end of the topic, a student should be able to know what is membrane potential, what is diffusion potential, what is Nernst potential, what is resting membrane potential, and what are the various determinants of resting membrane potential. So all these potentials, they simply define only one thing, that is membrane potential. And diffusion potential is a derivative, its potential is a derivative, and the resting membrane potential is again nothing but the membrane potential of a cell at rest. Okay, so we will see all these concepts. Please pay attention. It is very important for understanding the further concepts in physiology. Membrane potential means a difference that exists across all the cell membranes. That is called a resting membrane potential. So as we already know, cell membrane separates intracellular fluid from extracellular fluid. So a difference exists across the membrane of all living cells, and the inside is negative and outside is positive. So here inside of the cell is negative and outside is positive. So the potential difference is called membrane potential as ions arrange themselves along the outer and inner surfaces of cell membrane. At resting state of the cell, it is called resting membrane potential or resting transmembrane potential or simply resting potential. The term rest does not imply that cell is metabolically quiescent, but it is not undergoing any electrical change. Okay, rest means it is not metabolically silent. It is undergoing all the metabolic activities. Only thing is that electrically, we are talking about electrical potential. Okay, so electrically it is there is no change in the movement of ions across the cell membrane. So this resting membrane potential is different in different tissue. In a nerve cell, it is minus 70 millivolts. So how we got that minus 70 millivolts, we will see as we go through this further in this topic. When a neuron is stimulated, membrane potential changes and inside of the cell becomes positive due to depolarization. Okay, so how does the depolarization takes place? That is another topic covered in action potential. So this resting potential plays an important role in deciding degree and duration of the action potential. In our body, we are having two types of tissues, excitable and non-excitable. Excitable means they are able to give response to an electrical stimulus. Non-excitable means they are not able to respond to any type of excitable, that is electrical stimulus. So what are the excitable tissues? Neuron and muscle. So membrane potential, we are talking about the excitable tissues or excitable cells, that is neuron and muscle. They have more negative resting membrane potential, that is minus 70 millivolts to minus 90 millivolts. And these non-excitable tissues have less negative resting membrane potential, minus 53 millivolts in epithelial cells, minus 8.4 millivolts in RBC, minus 20 to minus 30 millivolts in fibroblasts, and minus 58 millivolts in adipocytes. So the basic thing is that excitable tissues are having more negative resting membrane potential, and these non-excitable, they are having resting membrane potential but they are not excitable. Okay. So the electrical potential exists across all the membranes of body cells. So these excitable tissues have unique ability to generate and transmit the electrical potential across its membrane, which is known as membrane potential. And in glandular cells, macrophages, ciliated cells, the local changes in membrane potentials also activate many of the cell's functions. So diffusion potential. It is caused by an ion concentration difference of two sides of the membrane. Okay, so the ions, concentration of ions on two sides of the membrane will produce an diffusion potential. So for instance, potassium concentration is great inside a nerve fiber membrane but very low outside the membrane. Because of concentration gradient from inside toward outside, there is a strong tendency for extra number of potassium to diffuse outward through the membrane, generating diffusion potential. Okay, so diffusion concept you already know in your intermediate. Diffusion means moving of ions from higher concentration to lower concentration. So the same thing here. Potassium concentration in this example, so potassium concentration inside a nerve fiber inside is more and very low outside. So what happens from high to low, the movement generally takes place. So that is concentration gradient. So the ions diffuse from inside to outside through the membrane, producing diffusion potential. Okay, so this is the one concept, diffusion potential, because of ion concentration difference on two sides of the membrane and movement of ions across the concentration gradient. So the diffusion potential caused by potassium ions is like this. The potassium ions carry positive charge to outside while moving, creating positivity outside and electronegativity inside the membrane, because inside negative ions remain behind and do not go along with the potassium. Okay, here you can see that as they are moving outside, they are creating more positivity outside, and inside, as the negative ions remain inside, anions, they create electronegativity inside. Within a minute or second or so, potential difference across the membrane, that is the diffusion potential, becomes great enough to block further net potassium diffusion to the exterior despite the high potassium concentration gradient. Okay, so after moving within a minute or second or so, the difference across the membrane, that is the diffusion potential, becomes great enough to block further movement. Okay, in normal mammalian nerve fiber, potential difference required is about minus 94 millivolts inside the fiber membrane, that is the diffusion potential of potassium. Higher concentration of sodium. So we have seen potassium, how much is the diffusion potential, and now sodium ions. So sodium ions, where they are present more in number, they are present outside, that is positive outside, and low sodium inside, assuming the membrane to be highly permeable to sodium but impermeable to all other ions. Diffusion of positively charged sodium to inside creates a membrane potential of opposite polarity, with negativity outside and positivity inside. Within a minute or second, the membrane potential rises high enough to block further net diffusion of sodium to inside, which is to be 61 millivolts positive inside the fiber. Okay, so till this potential reaches, the sodium from outside to inside movement occurs. Okay, so that is the diffusion potential of sodium ions. Thus, concentration difference of ions across a selectively permeable membrane under appropriate conditions can create a membrane potential. Okay, so the thing is that concentration difference of ions across a selectively permeable membrane. Their movement under appropriate conditions create a membrane potential. So till now we have seen the basic concepts of membrane potential, what is diffusion potential, and what are the movement of how the movement of ions takes place across the cell membrane. So now we will see the genesis of resting membrane potential and the factors underlying its genesis, or genesis means production, how it is being produced. Okay, so we will see these different so the cell membrane is selectively permeable to different ions. So what are the ions present? Sodium, potassium, chloride, calcium and bicarbonate and their concentrations. Sodium is more extracellularly and potassium is more intracellularly. Chloride is more extracellularly, calcium is nil inside and 2.4 milliequivalent per liter outside, and bicarbonate is 10 in ICF and 28 in ECF. So, sodium is moderately permeable, whereas potassium and chloride are freely permeable across a cell membrane. So the particle size of potassium is larger as compared to sodium, but the permeability for potassium is approximately 50 to 100 times greater than that for sodium. This is because of fact that hydrated potassium is smaller than hydrated sodium. As you all know, sodium is always hydrated, surrounded with water molecules. So that's why, though its atomic weight is less compared to potassium, it is moderately permeable, whereas potassium is freely permeable. So cell membrane is practically impermeable to intracellular proteins, that is anions, and organic phosphates, which are negatively charged anions. So presence of gated channels in cell membrane is responsible for variable permeability of certain ions in different circumstances. So for movement of all these ions, there are gated channels present in the cell membrane. So the next concept in genesis of rest membrane potential is Gibbs-Donnan equilibrium. So according to this equilibrium, when two ionized solutions containing ions are separated by a membrane that is permeable to some and not to others, an electrochemical equilibrium is established. Okay, and the electrical and chemical energies on either side of membrane are equal and opposite to each other. So in considering two solutions, each solution should be having electrical neutrality, that is positive charges should be equal to negative charges, and the product of diffusible ions on one side of membrane will be equal to product of diffusible ions on the other side of the membrane. Okay, so these are the two considerations under Gibbs-Donnan equilibrium. On one side, positive and negative should be equal, and on two sides, product of the diffusible ions on one side will be equal to product of diffusible ions on the other side of membrane. So if you take the potassium ions, potassium concentration inside the cell is more and membrane is freely permeable to potassium. There is an efflux of potassium takes place. So entry of positive ions into the ECF creates positivity outside and negativity inside, and outside positivity resists efflux of potassium, because since potassium is a positive ion, at a certain voltage an equilibrium is reached and positive potassium efflux stops. Okay, so as the potassium is moving along the concentration gradient, at one point at certain voltage an equilibrium is reached where the potassium efflux stops. In the previous slides, I have told you, till its potential is reached, that is diffusion potential, that otherwise an equilibrium is reached, the efflux or movement of ions stops. So after the movement of ions, sodium, potassium and other ions, so at equilibrium, sodium being greater on side which contain non-diffusible ions and anion is greater on the other side, their concentration ratios are equal. So there is an unequal distribution of diffusible ions, asymmetrical distribution. So intracellular fluid contains non-diffusible anions like proteins and organic phosphates. So according to this equilibrium, there should be an asymmetrical distribution of different ions, diffusible ions across the cell membrane, with cations being more inside than outside. But in reality, interior of the cell is negatively charged, which is explained in the further slides. Okay, so according to Gibbs-Donnan equilibrium, what it should be? Equal asymmetrical distribution of ions across the cell membrane. The next important concept is Nernst potential or equilibrium potential. The potential level across the membrane that will exactly prevent net diffusion of an ion. Okay, so what is a Nernst potential? It is the potential level across the membrane that will exactly prevent the net diffusion of an ion. So in the Gibbs-Donnan, what we have seen, there is an asymmetrical distribution of diffusible ions across the cell membrane in the form of excess diffusible cation inside into equilibrium, which results in concentration gradient. So the diffusible cations will try to diffuse back into the ECF from ICF, but it is counteracted by the electrical gradient which will be created due to presence of non-diffusible anions inside the cell. So an equilibrium is reached between concentration and electrical gradient, resulting in diffusion potential or equilibrium potential. So what is this equilibrium potential? Means that uh, equilibrium or the equal point where the concentration and electrical gradients are equal, resulting in diffusion potential. Okay, so it determines this potential. Nernst equation. What is this equation? E is equal to RT by ZF log inside or ion outside the cell divided by ion inside the cell. So whereas E is the equilibrium potential in millivolts of ions at which efflux and influx of ions are equal. R is the natural gas constant and absolute temperature. F is the Faraday's constant. Z is the valency of ion, and this is ln is logarithm. That is ion outside the cell divided by ion inside the cell. So at normal body temperature, converting from natural log to base log 10 and replacing the values of the constants in numerical, the equation can be simplified to plus or minus 61 log concentration of ion inside by concentration of ion outside. So the equilibrium potential for some important ions in mammalian spinal motor neuron is, for sodium it is plus 60, for potassium and chloride it is minus 90 and minus 70. So again, the diffusion potential level across a membrane that exactly opposes the net diffusion of particular ion through the membrane is Nernst potential for that ion. So this is one more slide for the same which I have explained you. Magnitude of Nernst potential is determined by the ratio of concentration of that specific ion on two sides of the membrane. Okay, suppose the Nernst potential for sodium, how much I have told you? It is plus 60. So how it is determined? It is determined by the ratio of concentration of ion inside and ions concentration of ions outside. So calculating with the help of that formula, that is E is equal to plus or minus 61 log concentration of ions inside and concentration of ions outside. Using that we can get the value. Greater this ratio, greater will be the tendency for ion to diffuse in one direction. Hence, greater will be the Nernst potential required to prevent additional net diffusion. This potential will be positive if negative ion is diffusing from inside to outside, and it is negative if the ion diffusing is positive. So when concentration of positive ions on inside is 10 times that on outside, log of 10 is 1. So that Nernst potential calculates to be minus 61 inside the membrane. And if the concentration of positive sodium ions on outside is 10 times that on inside, log of 10 is 1, so that the Nernst potential calculates to be plus 61 inside. So for this sentence, this is an example. So when the moving ion is negative, which is diffusing from inside to outside, the value will be positive, and if it is negative, if the ion diffusing is positive. So as I have told you already, the various potentials, Nernst potentials for the ions are like this. Sodium plus 60, potassium minus 90, chloride minus 89 or minus 70, nearer value, calcium plus 129, bicarbonate minus 123. So the Nernst potential is a potential level across the membrane that will exactly prevent the net diffusion of ion. And the diffusion potential when the membrane is permeable to several different ions. Okay, when the membrane is permeable to different ions, the diffusion potential that develops depend on three factors. Okay, till now we have seen diffusion potential or Nernst potential of single ion. But take a condition where the membrane is permeable to several different ions. So how do you calculate the potential? So the potential developed depends on three factors: polarity of electrical charge of each ion, and permeability of membrane to each ion, and concentration of respective ions across the membrane. Okay, so considering this condition, when the membrane is permeable to different ions, so this is the Goldman equation or Goldman-Hodgkin-Katz equation. So when the cell membrane is permeable to different ions at the same time, which is normally taking place in our body daily, so that depends on the polarity of each ion and membrane permeability and ionic concentrations. So here V is equal to RT by F log of potassium and sodium outside by inside and chloride inside by outside. So in the resting state, permeability of potassium is 20 times more than that of sodium. So from this Goldman equations, we can get various inferences like most important ions or development of membrane potentials in the nerve and muscle fibers are these three. That's why we have taken in the formula only these three ions. The voltage of membrane potential is determined by the concentration gradient of these ions, and degree of importance of each ions is in determining the voltage depends upon the permeability of individual ions. And positive ion concentration from inside to outside the membrane is responsible for electronegativity inside the membrane. Signal transmission in nerves is primarily due to change in sodium and potassium permeability, because their channels undergo rapid change during condition of nerve impulse and not much change is seen in chloride channels. Okay, and next moving to the ionic channels or the leaky channels. So these channels are leak means because they are permeable to the ions. So they allow free flow of ions. Potassium channels are large in number. Leaky potassium channels are large in number, whereas sodium leaky channels are fewer in number. Therefore, membrane is more permeable to potassium. So this sodium potassium leak channel, how does it play? What is its role means? A channel protein in the nerve membrane through which potassium and sodium ions can leak is called sodium potassium leak channels. So these are about 100 times more permeable to potassium than to sodium, because sodium is being hydrated and more difficult to move. So this difference in permeability is exceedingly important in determining the level of normal resting membrane potential. So if both are equally permeable, there is no chance in developing the resting membrane potential. So potassium is more permeable compared to the sodium. So sodium potassium pump is a very important uh, factor which helps in the development of resting membrane potential. So in the cell membrane, this pump is present which pumps actively. Sodium and potassium ions movement takes place actively, that is using energy from ATP. So it is an electrogenic, means producing electricity. that is 3 sodium move outside and 2 potassium move inside. Efflux means moving out, if influx means moving in. Net effect of causing negative charge inside the membrane, because positivity is going more and positivity is coming in less. So 3 minus 2 1, but inside of the cell is already negative because of organic phosphate and anions. So there will be negativity produced inside. So this pump causes large concentration gradient for sodium and potassium across the resting nerve membrane. So the ratio of these two respective ions from inside to outside are: sodium inside by outside is 0.1 and potassium inside by outside is 35. See the huge difference. So the potassium diffusion potential, how does it contribute to the resting membrane potential? So if the membrane is only permeable to potassium, the high ratio of potassium inside to outside, that is 35:1, Nernst potential corresponding to this ratio is minus 94 millivolts, and log of 35 is 1.4, and 1.4 into multiplied by minus 61 is minus 94 millivolts. That's how the potassium Nernst potential is calculated. Hence, if potassium were the only factor causing resting membrane potential, the potential inside the fiber would be minus 94. But this is not actually happening. The membrane potential is contributed by all the ions. This is only a theoretical point of view. Contribution of sodium diffusion potential. Assuming that membrane is only permeable to sodium, this causing diffusion of sodium through the leak channels. Role of ratio of sodium inside to outside the membrane is 0.1, which gives a calculated value of plus 61 millivolts. So sodium potassium leak channel is about 100 times more permeable to potassium than sodium. It is logical that diffusion of potassium contributes far more to the potential than diffusion of sodium. So using Goldman equation, net potential created by the leak channel, sodium potassium leak channel, is minus 86 millivolts on the inside of membrane. So the pump contribution, how much it is responsible for producing the resting membrane potential, means this provides an additional contribution to resting membrane potential by continuous pumping of 3 sodium to outside for each potassium, 2 potassium pumped inside of the membrane. With a continual loss of positive charges, sodium potassium pump creates an additional degree of negativity, that is about minus 4 millivolts on the inside. So the net membrane potential with all these factors operate at the same time, that is final value of minus 90 millivolts. Okay, so how this minus negative 4 came? With continual loss of positive charges, the pump creates an additional degree of negativity. Okay, so net value of membrane potential with all these factors is minus 94 minus, if you take 4 from this minus 94, it is minus 90 millivolts. So in summary, diffusion potentials alone caused by potassium and sodium would give a membrane potential of minus 86 millivolts, almost all of this being determined by potassium diffusion. Then additional 4 millivolts is contributed continuously by sodium potassium pump, giving a net resting membrane of minus 90 millivolts. So after knowing all these concepts, finally we see the factors contributing to the resting membrane potential. So this is just like a summary. So one of the main factors is potassium efflux. The Nernst potential of this is minus 90 millivolts, and contribution of sodium influx is very little, that is plus 60 millivolts, and sodium potassium pump causing more negativity inside the membrane, and negatively charged protein ions remaining inside the membrane contributes to negativity inside the cell. And the net result is minus 70 to minus 90 millivolts inside the cell. This is the resting membrane potential final value. So inside is so coming to the last topic, that is how to record or measure the resting membrane potential. We have seen by various equations how we got those values -70 to -90. But experimentally or outside the body, how to exactly measure this membrane potential? It is using microelectrodes and oscilloscope, which is about -90 to -90. So see in this picture, it is oscilloscope using the microelectrodes electrodes, which is one is kept inside and one is kept outside the cell, and another. So this is the picture of a cathode ray oscilloscope, which is used to record the membrane potential. So microelectrodes are present and the oscilloscope is present. So it is the instrument used to record the membrane potential. If we see the parts, it is having a glass tube with a cathode, fluorescent screen, and two sets of electrically charged plates. The cathode and anode, and the fluorescent surface, that is, it acts as a screen and electrically charged, arranged in two sets, vertical and horizontal plates. So this is the arrangement of the cathode ray oscilloscope. How does it work? What is the basic principle? The membrane potential changes during the activities of nerve fibers, which occur very rapidly, that is in less than a second. So the instrument should be capable of responding extremely rapidly. That means it must be inertia less. So first point is it should be uh, zero. It should be having zero inertia. And one more point is that the potential changes occurring in the membrane are in millivolts. So we can't record the millivolt change. So it should be amplified before being recorded. So one is it should be inertialess, and other point is the recording potential should be amplified. And these are the various parts, plates, vertical and horizontal plates. These are the electrolytes. This is the fluorescent screen. So there is no need of knowing the method. How do we do? These electrodes are present. One is kept inside and one is kept outside. And likewise, the recording or the changes taking place the cell are in the recorded in the form of a graph, and that can be seen on the screen. Okay, so this is the cathode ray oscilloscope recording of resting membrane potential. So we have seen the resting membrane potential in detail. So now I'd like to give few MCQs regarding this topic. Okay, so I hope you understood the topic and try to answer these simple questions. So first one is regarding resting membrane potential. What is the most important cation of intracellular fluid compartment? Okay, and next is what is the most important cation in the extracellular fluid compartment? How much voltage is contributed by sodium potassium pump to the resting membrane potential of a neuron cell? And what is the Nernst potential for sodium ion across the cell membrane while developing resting membrane potential? How much voltage is contributed by sodium potassium leak channel to the resting membrane potential of a skeletal muscle cell? So answer these questions in your notebook. So that's all for today's class, resting membrane potential. I hope you all understood the topic. So if you have any doubts, take your textbooks or refer this topic in the textbooks which I have told you, Guyton and Hall, Best and Tyler, Bernal-Levy. Try to search in the internet if you don't have those books, and please read the concept, the topic once in those textbooks, and you will be definitely able to understand this clearly. So thank you one and all.